Good afternoon, everyone. First of all, thank you all for coming to this a new platform which has been created by Institute of Chemical Technology in collaboration with Royal Society of Chemistry. This was an idea which was generated maybe 6 to 8 months back. And the idea behind the creation of this platform is nowadays there's a lot of discussion about startups, entrepreneurship, and how one could try to develop ideas which can be useful from the industrial point of view, from commercial point of view, and to a large extent more of humanity point of view. But we thought when this platform had to be created, what we understood as something which is a lacunae is many times whenever an innovator who could be an academician or who could be someone from the general public, we always expect them to present in front of us the type of work they have done, which always cannot connect with the people who are there. It's not always the science which gets connected with us. So we thought we could give a very free-flowing option, like a platform where a person who has gone through all the hurdles, the problems, and how they have overcome those problems. So starting from the idea which was generated, to having the startup, to developing it, and finally coming out almost like a commercial process, which could be helpful for many people. How that journey has been, if one can elaborate upon, can discuss upon very freely in front of the audience, that is something which we had in mind. So I would like to first thank the Institute of Chemical Technology, our Vice Chancellor, Professor A.B. Pandit, for supporting this idea. Definitely our head of department, Professor B.M. Bhange, who has been really very supportive. The time when I just mentioned to him, within seconds he has said that, oh, you have done it, then why not, just go ahead. Definitely Royal Society of Chemistry. This association with Royal Society of Chemistry has been very fruitful for us as ICT, because this started way back in 2012-13. It's one of the first interaction which ICT had with RSC for the Chem Career event. That was the first Chem Career event in India. And since then, we have done many events, not just in Mumbai, but also in Bhubaneswar and various places. So our Royal Society of Chemistry has been part of ICT, our chemistry department, for almost a decade now. And Mr. Rajesh Parishwad, which unfortunately has now has moved into the rival American Chemical Society, but he was the one, so I was discussing with Rajesh, and within seconds, rather, this proposal got approved from the RSC point of view. Mr. Ajit Sharma, who is the head of RSC India, he was supposed to come and attend this program, and unfortunately today itself he had some personal issues, so some problems there, so he had to go back. But I really would like to thank all of them that we have now started this innovation lecture series, and it's really a privilege for me. It's not because I know the speaker, Dr. Jayant Khandare, personally for almost 7 to 8 years now, but the type of personality what he is and what really he and his team has achieved in the last decade or more has been just an exemplary, what you call like a story. It's not even a story, it's a reality. What started off as a dream, but it is now a complete reality. So I just want to give you some details about Jayant. Jayant currently is the MD and Chief Scientific Officer of one of the company he has, he started, that's based in Pune. He also has a subsidiary in America. It's called as Actorius. It's mainly into research, mainly into biomedical type of research. And there is one product which they discovered was called as OncoDiscover. He definitely is going to talk more about that, but I'm sure everyone kind of knows that cancer is a big killer, not just in India, but all over the world. And one of the problems with cancer is when you have to detect cancer, in many times some of the detection kits which are available, or the systems which are available, kind of give you the information when the amount of cancer cells which are present in the blood or in the parts of the body are critical enough to give you better information or a better analysis. What his company has been working on is the ideology which we developed. You can have what are called a circulating cancer, which is present in any part of the body, but then the concentration might be very less in the blood, but you can still discover it, you can detect it very efficiently. And this, OncoDiscover is one of the first. I'm sure I'm not trying to steal his thunder. He's going to give the details of it, but this is kind of the first stage cancer information that is required critically for not just detection, but even for analysis and the further, improvement into the, the course of action which has to be taken for the patients. So this is kind of the first in India, but definitely not many companies in the world are also doing that. Just a brief background about Jayant. He has multiple roles, rather, I would say, over the years. So he's a chemical engineer to start off with. He is a polymer chemist, as a PhD from NCL. He then went on to do postdoc in America, in Germany. He was also an Alexander von Humboldt Experienced Researcher Award winner. He was working as a scientist in Piramal for many years, and I'm sure he will let us know why he probably left Piramal. But then there are challenges which are much bigger than just a job. That's what he always says. And he took up that challenge. After interacting with him, I know him pretty well, but today, after interaction, what I also come to know is rather than just developing some idea and taking it to a level where it becomes a company which is of use to people, he also has this philanthropist ideology that there are many people who do PhD postdocs, come back to India, and many times there is a struggle in life. Opportunities are not coming to us so easily. I could have been one of them as well, for sure. But then he has that knack of supporting people by giving them opportunity, giving them that stage where they can carry out cutting edge research. He's personally, through his support systems, funds them. And this science which has been developed over a period of time, which has gone on to become what it is now, these are the things which, these are the points which has really helped come out with all these options. So he's definitely going to talk about all these experiences. He's also a fellow of Royal Society of Chemistry. That is just one designation, but to be honest, he was holding the West India chapter. He was one of the main members. He was taking care of the West India chapter of RSC for 4-5 years. And that's where I started coming to know about exactly what Jayant is all about. I've seen him through his struggles. I have interacted with him on various levels, various times then when he was developing this whole technology. And today we are here to witness the whole aura of what his team, which is now around 75 scientists are working under him. And it's a huge team from, I think, 1 or 2 scientists who started off around 8-10 years back. So it's a big jump, and probably now the valuation of the company has gone up to maybe $10 to $15 million probably, or more than that, much more than that. So it's a big jump. And this is really what we wanted to bring to you all, that when it comes to the innovation lecture series, someone who started off with a simple idea, which probably many scientists in India and other way were not believing that it could happen. It was rather a pessimistic view which was happening on the other side, that it might not ever culminate. But taking up that challenge and coming out with such a prolific company and this, kit which they have created, which is now available in almost all the big hospitals around India. And to be honest, today when he has told me, I feel so proud when he said that in some of the big hospitals like Tata Memorial as well as AIIMS, for Indians specifically, he has made it free of cost, rather than the general costing, which is slightly higher than, let's say, 10,000 rupees. But he made sure that his commitment towards the country, towards our people, has been at the forefront. So without any much ado, I don't want to go further ahead. I think I would like to now allow Jayant to take the stage and give us a glimpse of exactly what he has achieved. But before we go ahead, I would like to, probably unconventionally, I would like to first give him a small token of appreciation on behalf of all of us, which probably tells exactly what the team as such has achieved. So if you may allow us.
Thank you. Thank you in advance.
Tries to use it for the benefit of people, humankind, I think that deserves an applaud.
Thank you. Thank you. Now I have a photograph. Thank you.
Thank you.
Go to other slides. So good evening, and it's a great honor. It's a privilege to be at this eminent institute who has contributed to nation in form of science and especially chemical sciences. Thank you, Dr. Anand. Thank you, your team, for considering me to be here today. I've been part of this dais a couple of times because of your earlier invitations, and that time it was probably not what we are today. But thank you to ICT, thank you to the faculty, thank you to the students today for assembling here and expecting something from me. But I have a disclaimer one, that it's all good to have such kind of a CVs. But if anyone is not as intelligent as is me here, I'm the least intelligent individual here. You are the most preeminent people with an capability in this institute. So I'm going to share my less intelligence capable outcome in terms of a translation of something that we started 10 years back, and I would like to narrate my story about a startup that convincingly started doing something which is often an impossible, as you mentioned. So that's a story that I would like to present. One of an objective, what Dr. Anand said, that some of you might want to have thought process and a catalyst, a thought process that probably is in your mind to have some startups. So I'm going to share my own story of my startup, but I will also share what does it mean in terms to be part of any startup, because we know that few successful scientists in this country who have actually translated something very meaningful, and not only India is proud of, but I think the whole world is proud of. So one of our personalities, Dr. Anji Reddy, I remember, biggest scientists, Dr. M.M. Sharma, Dr. Masharkar, Dr. Pandit. There is plethora of scientists who have contributed, and the next generation is sitting here. So, with all this acknowledgement, I stand here today to share my own story. And it might not be a perfect, it's not a perfect, startup story in the world of Elon Musk and, in the world of Amazon or in the world of an iPhone kind of a story, which are game changers, which are actually contributing in such a mass, which is billions of dollars and billions of valuation. But what it means to a startup is that one compelling thought process which will impact into human life. You might have cars, you might have something which can go to Mars, you might have something which is in the virtual world, which is important and which is actually creating impact to human life. But what is most important challenge that humanity is today facing is a healthcare problem. So, and in healthcare problems, it has been, I was just speaking to Anand, that if you heard about Dr. Robert Langer, our own Dr. Samir Mitra Gotri, who have 400 and hundreds of patents being filed, hundreds of companies, and they are all chemical engineers, by the way. My own first postdoc professor in US was a rheologist, is a physicist, but he today is a professor at Johns Hopkins, doing something into cerebral palsy, and a disease which is going to be very difficult to be treated. And so the boundaries, being a chemical engineer doesn't mean that you cannot contribute into biological sciences. But often those boundaries are actually built up, that I'm an organic chemist, I'm an inorganic chemist, or you are a chemical engineer. So I come with a little humble background, and I would like to talk about OncoDiscover, which is a circulating tumor cells technology, and why this kind of a technology is important in a modern world. So number one point, that even if we have great industrialization, we would have a greed of actually manufacturing and designing and having everything that actually is running into the next generation level of expectations. Our generation had different challenges. Your generation is going to have different challenges. So first point in a startup is that when you start something, you must have your own philosophy. What is it that you want it finally? Whether you are doing it for humanity, society, you are trying to solve a problem which is incremental, or it's just an me too, or an unmet need. So first you have to define where you want to go it. So for example, developing a new car with a battery car is probably essential, which will change the way this planet is. But somehow that change is so implicative that whole environment probably could be of a beneficial factor. But for Indians, for audience here, you need to realize that when we come to say NCL Pune or IITs or UDCT, you are on actually some obligation, because that's what an obligation that bought me is. Ultimately whole of my education has been funded by the taxpayers. Maybe I am a today taxpayer, probably highest taxpayer proudly here, but at time I couldn't pay even a tax. I wanted to save tax, but all my education has been funded by taxpayers funds, zero cost. Now when you complete your PhD from NCL Pune with Dr. Masharkar's lab or Dr. M.G. Kulkarni's lab, there is always an obligation for you to go for a postdoc, right? You want a job. Yes, of course you need it. But in academics, we never actually discuss that where you want to end up after 5 years or 10 years, or how do you want to retire it. Because most of the professors here, right, believe in a research, and we believe in publications, we believe in creating IP, we believe in actually, having an inductions, jobs. That's essential. But why do we do a job? That's more important. And how do you do that job is very critical to think on. Why do you really want to start up? That's a story I want to actually begin it. So don't think that I found a problem and I started my company. I did something because I am obliged to this country that who gave me an education from zero standard to whatever I invented. So my obligation as a scientist is towards a society, and which part of a society is the bottom of that pyramid part of it. But innovations in today's world, especially as complicated what I am going to talk about, liquid biopsy, is immense challenging. And that's a story that I will tell you, and why did I start my As Anand mentioned, that I was working as an innovation head in Piramal Life Sciences. After my postdocs, I had a job offer. And fortunately, I never applied for any job. I don't care much of a job. But because of a scientific background, and because I was in the US, Piramal wanted somebody of my kind. Either I have a job or I don't have a job. So luckily, I was in a part of that niche area where Piramal, somebody found me, and then, I came to India. I realized in 11 months that this is a great job, great company, great objective. And it was doing a drug discovery. For some reason, it started, very unmeaningful joy of doing a science. And one fine day, I applied to Humboldt Fellowship. And next day, I got an offer to actually join a Berlin. So we are very good as a scientist. We are wanted by, other forms of researchers. And I went back for an Humboldt and etc. When I wanted to come back after 1.5 years, I didn't have again a job. And then I preferred some private university jobs in Bombay, deciding that I would never, come to the government institutes like this, because it has its own greatness, it has own strengths, but it has own, you can't leave a job when you start something here, because you are tied up for life, right? Because your institute, takes a decision, and you have decided to serve that institute, because it's a national institute. And I'm not of that kind, because I want to actually run away from one job to other job or become an unemployed for some purposes, etc. After I rejoined Piramal, I realized that there are many returning Indians of a great caliber, intelligence. Some of them passed out from UDCT actually, went to great places, Harvard, Cornell, you name best of the institute. But the dynamics of the job changed in 2010. And so while I was actually inducted in a good company by an offer, by an invitation, in 4 or 5 years, since we accumulated all the positions, there was no positions for any great scientists coming to this country, even not for the Nobel laureates, for example, because of limited spaces that we have in. And in that 3.5 years of a journey, second tenure, I thought it is meaningless to be a scientist, whether you are Humboldt Fellow or whether you are Royal Society's Fellow. It doesn't make, because we don't create job. And I am very wrong to tell ICT students to create jobs, because there are hundreds of students that ICT has cultured, which has created jobs, created industry. Your Vice Chancellor's office, we saw, he was in a meeting where there's a big industrialists that were passed out from UDCT or ICT for that matter. So this institute actually cultures that kind of seed, which is entrepreneurship, right? At least highest number probably, if you see per capita per students other than IITs, maybe from this institute. So the advantage that what you get at such kind of an institute is already high. So therefore, when you want to do something, you must realize that what is it that you are solving it? Is it an unmet need? Is it required, right? Are you going to, survive for those 3 years, 5 years, or 10 years in a race of, value of a death? Because as a startup company, you always have a limited funds. And on the other side, you are actually sacrificing great jobs, great perks, great, positions, etc. So point number 1 here, startup is a sacrifice. It is challenging. Although it looks very fancied initially because you got funds from, say, national some institute like BIRAC or DPT or whichever agency. And our agencies are great, by the way. They give you funds if you have an inventiveness in your mind. If you have something novel, if you have something which is incremental and which is actually required by the society, you get great offers, and the funds are actually huge. Now, the point I'm trying to say here is, when I looked at this job towards my company, one fine day, towards end of my, second time exit, I stumbled upon one of a paper that came in a science. As an innovation head, I had actually freedom to do anything, come anytime, go anytime. That freedom was there, that liberty was there, that unboundness was there. Maybe I was only exceptional scientist in that company. Because as an innovator, you cannot be bound by rules, right? You cannot have a biometrics. You have to have a freedom and multidisciplinary team. So one fine day, I found a note that, what is cancer's problem? And the cancer's problem is in a circulation. And that is actually through the blood cells. So I read it, it inspired me, and it had a material science, and I posted it to my chief scientific officer, who was probably traveling worldwide. He used to come, once in a while. After 10 days, his secretary called me. And this is true incidence why actually I really started my journey in circulating tumor cells. So when he calls you to the office, there are two reasons. Either he will fire you, or he will, give you something incremental. That's a very straight way. And my thought is always that to exit. I never like to actually, be there and retire like Anand probably would do here. But I am always on the hot seat. And I went with an intention that today is going to be a last day, because this was 3 years already and time to move on. Earlier I moved in 11 months. So he came and I went to his room and looked at, he said, Jayant, you submitted something to me. Very nice paper. I said, in last 10 days, so many things I have read it, right? Currently I am watching actually India-England match. He said, you are watching actually India-England match in office? I said, I have nothing to do today. So I was actually reading Times of India and Creek Info, etc. He said, you should do something like this. And I saw that he was actually printing that paper, read it from that bold title, and I actually escaped from his room, went back to my office, started reading again Times of India, without actually uttering a word. His secretary calls. She was obviously very upset. And he said that, where did you go, Jayant? I said, you were saying something, so I thought it was not important, so I went and started looking at the cricket match. We are badly losing, by the way. He said that, you should probably get into this kind of a technology as a material scientist or chemist scientist or polymer chemist. I said, I'm not a biologist. I'm not an oncology expert, although I have a background of molecular biology later on in US, etc. But I said, okay, let's have fun. I have 3 conditions. So, he was a very wise person, and as a CS of a company, he said, you are putting 3 conditions on me, right? I said, okay, let me hear out, he said. I said, first of all, increase my salary to 1 crore per year. That's good enough for him to actually say, get out, okay. So second condition, he says, very interesting. Second, I said, I need 100 crore rupees budget. He said, get out, okay. There's nothing like that 100 crores. And I said, I need 10 years of undisturbed way of my working. He said, that's not possible, right? I said, if I had capacity to make technologies like this with this peanut salary, right, in this company, with all liberty and freedom, and with all this company of scientists here, the way we work is absolutely absurd. So therefore, I would not take your 100 crores, 1 crore rupees, and therefore I would not actually want to dare into this system. Fortunately, for some reason, incidentally, we are working on 2 materials, some material magnetic nanoparticles, and we found that these materials, because we could have some protein, it would have a great interactions with these cancer cells. And we published a paper. It came as a, inside page of advanced healthcare material. And one fine day, 3-4 months, I think I quit, because I was so frustrated that many scientists came for a job. So twofolds how I stumbled onto the circulating tumor cell is that science paper 2010, which said what is cancer's problem. And I'm going to actually cover that as we go. So it is not about, a company giving a job and actually asking you to do it. It's impossible to translate this kind of a technologies single-handedly, even if you have 10 crore rupees or 100 crore rupees in that structure. It is a time, it is a destiny, it's a kind of an incidental that you might succeed. So the success rate of this kind of a technology is below 0.1%. That's what I pitched to DBT, that when they asked what is the success rate for this technology, I had written actually 0.1. And the chairman said that, this is so high risky, because BIG grant is a high risk innovators grant that I stumbled upon. I said, you are not looking at that 0. After 0, there is a ballpoint dot over there. and still committee actually trusted that we would actually, do it. So that's a story about actually a background of why did we start it. So if we look at what is the recipe of startup companies capacity building as a content in my talk, that you have to have a 4 E's. And these are very important because you might have a great scientific capability, some idea, but these 3-4 E's are very, very important in my, my point of view. Ethics, the way actually you will practice the science. The ethics, the way you will keep a startup. Because later on you are going to meet to the angel investors and the VCs. They are very smart. They understand, you are cutting corners over there. So scientific ethics and your human ethics are very, very important. How bold, how straight, how, great you are about actually putting this is the finest line I feel is important. Educational capabilities are definitely important. If you come from ICT, you come from Stanford, you come from Oxford, you come from NCL Pune, it creates a credential of any organizing body who is going to fund you. So educational capabilities are definitely important. Of course, some dropout people can also make magic in this world. Steve Jobs, for example, right? Or Stanford dropout. Or we should have a trend of actually ICT dropout. You don't need 200 plus PhD students completing their PhD and getting. If you have really thought that you don't have to be, in a completion of a degree. But educational capabilities definitely builds your possibilities. Experiences are very important. The one technology which I am mentioning required actually 15 to 20 years of transitions from one lab, one country, one center, one academic place to other places. Without that, you cannot actually have something, as challenging as this. Experiences are definitely important. They should be packed up into your bag. Expectations. What are your own expectations? Like you are working against 99% probability to fail the technology. Why you want to actually still, drive that story in a 1% probability of success. So you might have a safer pitch which will have a higher success probability, or you might have a tendency to take a challenges with actually only 1% of. It depends upon your own individual, capacity, your mental ability, your thought process. And therefore, I think these 3 or 4 E's are very, very important. Now, startup has several challenges. And important, in this country, we always talk about the frugality, right? Everybody around me, people that I mentioned earlier, they always want you to do something meaningful for the lower strata of the society. That means 10 times sensitive and 10 folds lesser cost of it. Worldwide, no startup company, especially in the Western world, will have this kind of a philosophy. So you need to actually understand, what you are talking about a frugality. Frugality doesn't mean that you can just simply, waste your resources. And these resources are very, very important as you go ahead. Because, scientists who want to join you are not going to, believe your frugality model. Because they have done PhD from, Cambridge. Why should they join your objective? They are looking for a job. Anyone in this auditorium today just want to come and work with me without actually expectation from a salary. That doesn't happen. Therefore, my model of my company is always to give a job those who are really desperate, not to give a job to somebody who is great, who has experiences and has an highest salary, somebody that can pay. We don't do actually that our company. And that's how the startup companies cater the capabilities of human intellectuals in this country. And this country has many, many such students who need actually, jobs when they pass out. Remember, focus with an uncertainties. You know that why you are going to fail. So you have to actually daily, keep a note, at least in mind, that what is your tomorrow's challenge and where you are going to fail. So those uncertainties actually kill you every day, every night. Because if you are 35, 40, 50, right, you have your personal responsibilities, you have family, and everybody is going to actually ask you question, when is your technology coming? When are you going to actually come in a market? When are you going to be richer, right? Especially if you are a wife, your husband is going to ask, and if you are vice versa. Everybody is talking about your actually success. They are going to build anxiety. So you have to actually tell them to shut up for several years. That until you say that there is going to be a success, they don't ask. And thankfully, my parents, they don't understand what I do, and they don't care, but they are happy that today something is actually coming over there. So your own, expectations are very, very important. Don't live in the world of your families and your high peers' expectation. As a startup, it is your own dream, it is own challenge, and your own success later on. So what we have done so far, we started in 2013. We are in now 2022. One American company actually scouted us and said that, let us do something in genomics because you have a history of creating such kind of medical devices in this country. And offers that actually come at your doorstep. And you don't want to take it because you are actually starting another decade-year, journey of challenges. And it becomes, you can't run, you get exhausted. So startup is a marathon. So better start running marathons. That's what I did actually when I started doing my little serious science. I started running full marathons for 4 hours, 5 hours, out of house, out of offices, out of friends, no phone calls. And then your thought process actually starts of a vigor. Your endurances, physical endurance is very, very important for your journey of a startup. If you are unfit, if you are actually not capable of actually standing several hours and running and travel, your journey actually closes. That's what my one of a take-home message over here. So today we are actually in a circulating tumor cells and a precision oncology company. And I'm going to tell you what does that mean. So we have done several breakthroughs, first time in this country, second time in the world. And we are leading company actually what we do through our IPs and regulatory paths, including into big hospitals like Tata or AIIMS hospitals, etc. And we are good in actually now commercializing this technology for the masses, if not for the reaches. So that's what is actually most important for us. So stay away from, excessive funds. Even if you have if you have a lot of money in your pocket, there will be no driving force for you to go to your startup. So it will make you lazy. It will actually, detach you from the success or take you away from the success. It's my philosophy that board of directors, advisors, especially those who have not done anything in their life, but they are good academicians, go to them, touch their feet, okay, understand what not to do from them. That is what is most important, right? It is important to be with, high peers, right? I work with my PhD guide, although it's like almost now 20 years that, he's a student of, again, incidentally from UDCT. And we I like to meet. Yesterday he came to my office and said, Jayant, this is a book that you should read it. Gift of an Athena, that's what I am reading now. And I understand how olive tree was very important to this, one of an European country. So learn from your advisors, but don't take them with a baggage because they will drive your journey. And the journey which they have not translated, you should stay away from it. Stay away from the media exposures. We have very, very small media exposures. I don't apply for any awards, and I'm very happy that somebody like a Shark Tank or something, they called me, I switch off. They say that, I'm a Shark Tank, you want to come. So I said, thank you for call. And again they called. We got some, I'm giving invite, special invite. I said, thank you, switch off. They said, thank you, but you are not actually want to talk about it. So stay away from, some such kind of situations, which is also important for your exposure of a company later on when you, travel a path. You need this kind of tractions. So team, technology, and traction. These T's are very, very important for you. But the tractions, which are actually media, they will call you, you will get awards. And I know at least 10 to 20 companies around me who actually are surviving on the awards, cash awards. And they are losing focus actually why they started a company. So it's really not good to get those kind of exposures. So what we have achieved, OncoDiscover is first in class, first medical device that Indian regulatory body called as Drug Regulatory General of India has uploaded. And this is very humble success. I would like to present it here to Anand. And thank you very much for calling me here at ICT. And it owes 10 years of hard work that actually goes to the team. So, what this OncoDiscover technology does is indirectly or directly saving cancer patients' life. And that is what is actually important. Your startup and that meaningful impact that is compelling to the human life, especially to the dying patient, is what we do it. We save lives. And that is more important than any other success, especially the financial success or valuations. And I hope I, put it here as a summary what we do. We impact cancer patients' life by increasing their survival. And only God or destiny has that capacity. But today's modern science, technologies, innovation has more meaningful capacity than actually a creator of this, life. And that scientist has that purpose. And therefore, that obligation to do something is very important, that can we impact patient's life. And that is what, you discovered new catalyst, that's great, right? But your material that was a drug, if that material was important for creating something great, that is what is very, very meaningful. So no science is less or more. It is your thought process, how you want to actually lead your science journey ahead. And I will actually share what are the cases in real world. I will have some disclaimers over there if you want to see why do we dare to say that we can save patients' life? Why we have actually that capacity? Why science has that capability? Why that, interdisciplinary group can do it? And that is what is the purpose. I think you wanted me to talk about, the content of a startup. I think I have sum up here what is a startup, that meaning. So I will, shift my gears to the second level. If anyone has a question at this point, I will be very happy to take it. About startup or about, I have interesting stories ahead. If there are not, we can stop here.
Any questions?
So far, so good, ma'am. Right? So, if you have not seen this cartoon, this is very famous. This is very interesting cartoon for me. Anyone has seen this cartoon earlier? Raise your hands, please, if you have seen this cartoon. You have seen it, great. Youngsters, anyone has seen this cartoon? Put yourself where you stand here today, right? Typically in science, okay, when you become graduated from undergraduate to PhDs to postdoc professors, we have, different thoughts about each other, respect about each other's science, right? And very interesting, cartoons that you should always also have, because your journey as a startup should not be boring. Many a times, because we are professors and we are actually, such great academicians, which is important, but we should not convert into a boring personality. You always need some humorous, right? Some unusual, humors are important, because people around you, they can't be sticking to you because you are serious of a science. And if you see here, I would ask actually Anand, which one you liked, or sir, which one you liked it here, or where you actually narrate yourself or correlate yourself, right? Right. So, yes, one professor to other professor probably are receiving a Nobel Prize, right? Maybe Tanak or somebody over there, right? And we always feel that, somebody is enjoying in their life far better than you. So this is all journey of a scientist who start up their companies, and they would meet each of that kind of a character, undergraduate, postgraduate, PhDs, postdoc, better scientists than you. They will actually be far superior to you, and you need mixture of all these people. And if you have that capacity as an individual human being to work with everybody, even with your own student or an interns, you have actually very positive outcome whatever your goal of a startup is. The problems with scientists is that we have our own space, physical, intellectual, and whatever that we want to do. We are often not open. We don't express, right? Because we feel that being an ordinary is going to take you down, right? Often that happens. So culturing actually a capacity, because if you are in a great institute with 4 years or 5 years of a PhD, there is a lot of burden, and it changes. And I have seen that happening to my own friends in NCL and other places. So that personality, it has to be, very interesting. Because when you want to meet to an angel investor, I met my investor on a phone call, and he said, Jayant, I have only one question. Why do you want to do what you do? I said, there is no concrete answer. I am an egoistic guy. I want to have actually esteem that somebody can do this in country. So I am going to make my country proud. That was my statement to him. He said, what do you think about, what is that valuation of your company you expect? I said, I never understand. Arvindan, my colleague, understand this business or, marketing or sales. I said, I am here to show that my countrymen scientists that we can do something in this country for these people in this society. So that's an esteem. And if I succeed by 1% of a chance, I would like to donate this company to anyone that you advise. He wrote me a check, and I said that 2 years you should not meet to me. And he did not meet me for 2 years. In fact, when I went to US a month back or 15, 20 days back, he showed me a sign saying that, Jayant, I have actually put my house on a mortgage and selling house for providing funds for you. So angel investors, they are also human beings. Even if they have funds, they are judging why you want to do it. If you say, I want to be richer, absolutely it's fine. But be with a conviction, right? Why you are actually want to do it. And all of us probably have different questions. Don't have that modulated thought process speech, which is very, very boring. Don't get in. Be original, because that gives you an impulsive, intense context to the question that somebody asked. So that's very difficult to do it. And if you have that originality, I think that goes. And then you can have your co-founder like Arvindan Vasudevan. You can meet Dr. Pankaj Chaturvedi, one of the best oncosurgeons that this country or world has actually in Tata Memorial Hospital. You can get awards. They invite you. They give you, global innovation awards, right? Without actually putting an application even for a minute, I think they called, and your central minister can actually start your company. Most important is that you are in a company of an oncologist. Never ever in India that there will be a polymer chemist or chemical engineer sitting in actually a conference, although you organized it or you are actually at other places, will make a space for you, physical space to sit you. Because their own society doesn't actually accept it. Like our society doesn't accept actually other forms of sciences. And this is what is actually I am trying to say here, that if you succeed, I think, many great things will follow you up. But you have to actually have this thing on the board, which is very convincible why you want to do it. And that space I am very fortunate to actually accommodate with oncologists who are taking care of those dying patients. And CRISPR technology. One of my PhD students in 2020 gave me a book which is called as Code Breaker. And we were all in the COVID, and everybody was sitting at the home. Because we had a technology, we used to get blood sample. I used to go to my office and I started reading that book. So CRISPR Technologies scientist Dr. Jennifer Doudna, all of you heard about how the genome is going to change. The first line of this book, which is a code breaker gift by my student, I read only one sentence and stopped it. And the reason is the first sentence said, a Nobel laureate of 2020, her own life in that COVID time says that Jennifer Doudna could not sleep. And I said that let me close and guess what does this statement means. If you're a Nobel laureate, you would sleep and never get up, right? Or you would, have every day some kind of an event. So I was wondering why Dr. Jennifer Doudna is actually sleepless on date, which was 22nd March 2020. So remember, your science finally comes onto the ground, because COVID started. At Berkeley, she had to initiate COVID testing lab with 100 people around her, and it was the first institute in world which actually offered COVID testing at Berkeley. And that is why the Nobel laureate like Jennifer Doudna cannot sleep it. I hope all of us do sleep every day, right? With achieving and without achieving. Tell yourself as a startup company, you actually have to sacrifice such kind of things by writing, by doing it. You cannot go to sleep with an incomplete work. The purpose of working from Monday to Friday, right, or for 5 days, is not about a week that actually you are getting into. You should not know actually which day it is going on, what date it is going on, right? As a startup company, be ready to work 24/7, right? That's what is a startup, innovator's journey. You don't have time. You have freedom, but you have actually all the work in the world. And I meet, as I mentioned, my first professors, and the person who actually, the doctor who coined a word called as a liquid biopsy, I keep him as an acknowledgement that I met him very recently, Dr. Klaus Pantel. And in a conference of 22,000 people, a conference called as an American Association of Clinical Research, I wanted only one person to meet. And that was actually Dr. Klaus Pantel. And incidentally, I met him and offered him a lunch. And that's how actually we went to, other places. You want to meet that person, you want to actually convert some science into a technology, that will definitely happen. And probably many people will help you, but a leader is in you. And you are on a driver's seat. Don't let that be controlled by others. And that is what is the most important context I am trying to mention. My 20 years before relations with all my professors, you need to spend time, money to actually go to these people and share your journey, stories that will inspire you, because all around us not necessarily will have an successful stories, right? We might have a mixed bags of stories of success and failures, right? Reach at a success stories, knowing that there is also going to be a failure stories over there. Next slide, please. So this is my American company joint venture called as an OneCell. So we work in a single cell, single circulating tumor cell. We work in world's highest genetic mutation profiling called as a comprehensive genomic profiling of cancer patients today in Pune. And nobody is as good as what we do through artificial intelligence that this company offers it. And this is right happening in actually Pune. So you seed something, and then you actually get a plants, or you get big trees ahead. And people will scout you, and then they will offer actually funds to you. That is what is the story of my team, including Mohan and Dr. Gohar over there with Arvinder. And it's very interesting and very challenging. And you can create further jobs and labs like this. So next slide. So there are many risks and problems. What we spoke in first slide as a content, yes, there are risks and problems, and there are also challenges associated with, but then you and your research is very, very implicative. And I understand that, all of us are aware of actually challenges of a startup companies, where your idea is something, and then you actually, move into many, many trajectories. And either you be blinded about it, knowing that there are 100, steps, right? Many of you would not dare to take those steps, because today my intent is not to frighten them for not to get into it. But either you take each day, each step, at once, ignoring what is challenges ahead, or you fraction all steps in advance. So you are aware that what is going to come in. And if you do this mix and match, sometimes you need to ignore, sometimes you need to be aware, so that your success in your risk can actually go down in the startup companies, as this picture says. My team actually keeps on building. So anyone who least work today in company is me. Rest is actually my team. So acknowledging my team over there who does wonderful work. Otherwise, spending a whole day here is only worthy because friends like Anand and madam and meeting new people and student is only the worthy. Otherwise, there is tons of work that actually you are supposed to be. And it's a good situation that you don't have a time. But I told to both of them that I have a lot of free time when my friends call me, especially ICT calls me. And this is my 4th time, I think, coming on this dais today. So you have to be available for next generation of people, next intelligence that this country has. You can't be selfish that you succeed and then you travel your path. It's again an obligation to the next generation. And thank you, Anand, for actually inviting me here today for that purpose. Next slide. So, is there a market and a need? Please ask yourself. Unlike me, I knew that there is a need. And after 10 years, probably there will be 10 companies, 100 companies who will be offering this. So, you will be not be there. But I was pessimistically sarcastic and optimistic that nobody in this country is going to invent this. So I was, either pessimist by that virtue, or very confident that nobody will do it, and took as a responsibility that it is me that has to be done. Me and my team actually could do it. So please find out your technology innovation space, material, product, whatever you invent. Does it has actually a necessity to bring onto this earth? So that's very, very important, and it will come incidental, as I mentioned. You read one paper, as a material chemist, scientist, you can then design any complex material. Any form of a catalyst, Dr. Anand can actually easily crack, right? But only the problem is that, what is that your chemistry of an utility? What is your physical sciences for a capability of, right? And not all of us can be a translational scientist, by the way, right? But there are definitely possibilities of this new world. Now, what is more important? It is not about you and your idea, right? For a moment, you consider there is a fantastic concept and concept that has been formed. You have to actually marry your co-founder for 10 years, not in a literally word, but one of my co-founder, my co-founder Arvind, he always used to come to me in my office and say, good morning, sir, good afternoon, sir, good evening, sir, kya chal raha hai? And we met on actually marathon, we used to go to Goregaon, not Goregaon, this Sanjay Gandhi Park, 5:30 on Sundays, because I had bug of running and he also had bug of running. And since I ran on one of a Mumbai marathon, he heard that there's a new scientist that has come and ran a marathon, and he started bugging me about marathons and running, etc. And after 3 years, he said, Jayant, you should start a company. I said, I'm not made for it. I don't understand business. By the way, if I have convinced you, I don't understand a business till today. But he said that I will actually want to join you. Sacrifice. 10 years, 24/7, actually Arvindan Vasudevan works for the company. Ignoring health, ignoring life, ignoring personal challenges. So somebody younger than you or junior than you comes and actually opens your eyes, throws water on you. And we are so stupid as a scientist, not knowing what is our capability. So you need to have, those kind of people, youngsters, friends, colleagues, relatives who can actually tell you that you have that capability. Like Arjun was told by, Krishna, probably you have your teacher in front of you. And the whole purpose of your teacher was to make his job meaningless. And that you would not realize it, as an Arjun maybe, right? Somebody will actually tell you. And that circle you need to have it. Without your co-founder, you are nobody. Without your team, you are nobody. And that you need to realize it. So that confidence, that component is very, very important psychologically to imbibe very early. And most often we say it's me, it's my science, my experiences. Later on you realize philosophically you are actually one small part of it, of a journey. I think I'm going to talk about now, the quotients which are important. So, and there are 4 or 5 quotients I have. Everybody is intelligent here, right? Is there anyone less intelligent than me here today? Please raise your hand. I already told that this is the most preeminent institute, right? So I'm very humble to tell you that nobody is as dumb as me. If I can do it, you can do it. So your intelligent quotient has to be optimized by you. So that's what is the foremost important factor for you to actually consider when you start up anything in a journey of your life. Is an emotional quotient. You might be intelligent, right? You might be a superhuman being. But the problem with this superhuman being, they might not have an emotional quotient. And this is very important because you need to be naturally with an emotion quotient, or you need to derive and articulate to be an emotional, because it is required to express that what is actually, to be nice. You can't be a Steve Jobs. If you have read Steve Jobs' book, you will realize how stubborn he was, how not good he was actually understanding, what is the purpose. Later on, because of various things, that he had an emotional quotient. Now, most important is N, and that is a network quotient. Initially, you thought that you had everything in place. Later on, you need friends, who can help you. So naturally, the scientists do not build their, the network quotients. We are very poor in it because we are shy about creating communications and building it. Every network, I realize it, how important it is. And one of the reasons why I don't, hire people by giving an advertisement, because whenever I need somebody in my lab for a particular purpose, the person actually comes and sits in front of me. Often recommended by other my friends, because your network is such strong that anyone wants, something to be done, they send you. So you save your energy. F is, financial quotient. It's very, very important. You need to survive. That's the main game. A is, I think, more important as a startup company, that adversity quotient. If nothing works, what are you going to do it? How are you going to come back? They say that fail fast. But I must mention that inside me, if there is 1% success rate, I follow a mission which is called as failure is not an option. It's very good to say that fail fast and recover. In general, if your startup company fails after 2 years, if somebody has funded you, including government, do you think that you will actually get another investor for the same idea? So failure is not an option. And that is what is a complete philosophy that we want to actually do. You can be a professor, scientist, which I am for last 11 years, right? Keep it low. When I go to academics, I just talk about what we publish and how it is important for a science, how it is on the cover page, etc., etc. Build that story, publish it as a cover page. And this is very important because people with exceptional, ordinance of skill in the art, they should know that you have something in it. Remember, say you met a 2 organic chemist or 2 catalyst chemist. What is our version about somebody being good, better than you? We always are in our own, phobia, own thought process that I am better than you, right? That's always, a problem. But investors, which affiliation, where is your PhD, what is your experiences? And investors, they like people who publish papers. They like people who file patents, because that tells your seriousness about your science and about your capability as an innovator to create novelty in terms of an inventiveness, in terms of a commercial utility, in terms of an expenses that you do into a patent. I must mention here, while it is very important to innovate in a region where we are, unfortunately the laws of US and India and Europe are very, very important, are different for filing a patent, right? So filing a patent is important, but how do you protect and how do you continue to survive your patent? I must mention a US patent, PCT application, Indian patents, combiningly will take at least 10 lakh rupees on day one if you really want to protect your intellectual property. So stay away from filing patents if you do not have funds. Fortunately, I was telling that, we have filed now several patents. Every alternative week, your attorney will send you some FERs and examination and then bills. And then you will start actually feeling, why did you file this patent? So much of a money, $40,000 money fees will come. So it's not fancy to innovate. Stay away from it if you don't have enough funds. But if you really want to get away, yes, publications are very, very important. Series of, your cover pages will come, with some impact or, some meaningful impact factor journals. Go ahead, please. So here we started in 2030 with Arvind and Vasudevan. We have several patents and we continue to file patents. And we are India's first medical device company with ISO 13485, which is a global harmonization quality management system essential for medical devices. It's very, very important. We are today world's finest company for a job that what we do in terms of sensitivity, specificity, and accuracy. And this is not because I am saying, this is what most of the publications and clinical trials are talking about and doctors are talking about. So We got an advantage of this technology which was available in US in 2004, approved by US FDA. The cost of running this diagnostic test on cancer patient was $2,600 when we started. How many patients in this country can afford $2,600? Only 1% of the cancer patients can do it. How many times this test actually they have to do it? Lifelong, at least 3 times to 4 times in a year. How many of them actually will continue to do it? Their survival, especially in the metastatic settings, is very short, uncertain. And I'm going to cover that cancer biology as we go it. Whether you like it or not, I have to say what we do finally, right? Otherwise, they'll say that there was a lot of pop-up talks here, but there was no science that was disclosed. So I'm going to make those nerdy guys who want to actually listen science or like the biology of it. But remember, you can't actually develop a technology which is not affordable, accessible, and available in this country, which is, 3 A's that WHO inspires, that every human being on this earth need to have an access with the technologies which are there. But that's very interesting. No company in the world will develop a technology which can be affordable to the masses. You are in your own world. Even if you spend money and do this in frugality, finally, big companies are not going to like that your frugality. They will actually propose something which is going to be against your even sensitivity. So the balance of this innovation in terms of an affordability has to be achieved. And nothing is actually available to lower strata. 99% of the patient in India cannot afford my technology, which is only 15,000 rupees. And I feel bad for that only word over there. We want to do this at free of cost to those who do not afford it. And that's what actually, Anand, I want to do it in this for country. Therefore, my startup sum up here at this point is a social entrepreneurship for people in this country who needs this kind of a diagnostic, who needs to live for some days or their relatives need to expect them to actually survive it. And that is what is a mission that we are onto it. It is not a story of startup company with some product and actually doesn't have a real compelling impact into the cancer patient's life. And it's very, very challenging. That is what I think I want to say this through the slide. Look at the journey. Long. Most important is, your clinical trial journey is extensive. It might take a couple of years for you to get a patient's blood sample. 2 years. You might get, couple of years permissions from Drug Controller of General of India if that technology is non-predicate. That means there is no innovator in this country. There is no clinical regulatory pathway. There are no ideas. There are no concepts. There are no methods. There are no expert committees. There is no expert. And you still want to do it. That's why I said that this was impossible to do this in this country. Today, even if somebody asked me to give as much as fund to develop another IP for this technology, I will actually, go to Himalaya rather than running the journey. Because you run a marathon, you keep that energy to begin to your end, a step by step. And running a second marathon, anyone who runs a marathon here, 42 kilometers? I have run several. I have finished my 25th marathon. So I know that if you start running on 42 kilometers, you will actually curse yourself that why did you took this? And you never want to do this. But when you reach to the end with that endurance, I bet you want to actually register and run for the next marathon. So journey of innovators is actually one marathon to other marathon. And if you look at the extreme right, the abstracts that goes into this world congresses, if you do one abstract submission in AACR and ASCO, oncologists feel that they have achieved in their life. They have achieved nirvana of science that they do it. We had 9 abstracts in 2023. Couple dozens of abstracts from Indian company. We contribute 20% of science in circulating tumor cells to world in these conferences today. Sitting in India with our patient population here. That's what is a scientific contribution. If you want to talk about an impact factor of these journals, even with an abstract is more than 50, by the way, if you like that impact factor journal. And that requires hell of funds, hell of your capability to drive that clinical pathway. Next slide, please. You need, clinicians around you. And I funnily sometimes says Dr. Pankaj Chaturvedi, he advised me not to get into circulating tumor cells in first 1.5 minutes of his time. Don't listen. And that therefore he said, Jayant, when I tell you something, you don't listen. And when you don't say anything, you are definitely to do it. Because don't get demoralized. Why they are saying don't do it? That means indirectly they are making you aware what you are getting into it. That itself is a coach job. And Dr. Kumar Prabhash, India's finest oncologist sitting in Mumbai, Parel here. I see them how dedicated their life is all about. Every day they see actually patients who are dying, with an uncertainty. And that is really, human touch. Royal Society is all about human touch for a chemistry, right? Why we do a chemistry. That is the real contribution that your mentors, and we are very fortunate that these mentors are actually with us. We are doing something that offers to the world knowledge of oncology and biomarkers from head and neck squamous cell carcinoma. We have done world's largest clinical trial, which describes why actually Indian population with head and neck cancer patients die. Because this is India's cancer, it is human tragedy that actually we have bought it, because head and neck cancer is rampant in this country because of use of tobacco, smoking, cigarette. And it's a human tragedy. It's a human, intended utility. And every continent and, global spaces have different cancers. And you can talk to your science minister who will inaugurate your lab. They are there for a couple of minutes, right? You take a photograph as now it goes on. And they are on their way. They will say, great job done. And then they will actually move ahead. So this is a long clinical regulatory journey. Those who want to get into the biomedical sciences, talk to me. I will actually convince you not to get into this regulatory pathway. That is also an intention, right? But you want to get in, then probably let us discuss how does it goes, what are the challenges, right? And we need to be, this is one life, if we believe in. Use it, have a great purpose. And therefore you are in the elite institute. Most eminent institute, if anyone actually can be sitting today, is actually this place in this country. For the chemical sciences at least, right? Take that eminency into your head and take that opportunity to do something for the society. So I already sum up, this is what is actually the cancer patient's importance. If you give me 6 to 7 minutes, I would like to get into the third gear now. What is this technology for? All of us know that there might be many, many challenges, and we are contributing healthcare challenges as a burden to the several diseases by our physical presence on this earth. Forget about you have car or what emissions you generated. You look at the numbers that global cancer data is scaring. 18.1 million patients actually got into 20 million incidences, close to 20 million incidences in last 2 years of a time. This number is increasing. Cancer incidences are increasing. And there are 3 prudent reasons. One, and this is not my reason, this is quoted by the Global Cancer Lancet editor in 2018. The first reason is the increasing population with an higher age. Geriatric population is the main cause of actually increasing the cancer incidences, prevalences in this country or worldwide. So it's a natural process of actually deformations led by various biological factors. The second factor is sedentary lifestyle. Only I am actually standing and the photographer is standing. Rest of you actually are in the sedentary stage, and therefore do not practice a science which actually keeps you on the chairs all the time. So standing is very important. So sedentary lifestyle is going to harm us, and it's harming world. And the third important is a commercialization greed. Innovators are coming, starting companies, they want to do something, and then we want mass production, we want actually everybody to have great food and great human life. That is actually compelling this greed of commercialization, increasing free radicals in your terms, which is actually enhancing the changes in the human body, leading to the genomic changes and mutations that are happening here. So can anyone control geriatric population, aging of people? You can't. Can you control actually the commercialization competition. Our politicians, our scientists, they are going to continue to discover and create a burden over here by increasing the free radical. And probably you are not into it. Anything that you can prevent is a sedentary lifestyle. Food, habits. India is actually getting into colorectal cancer as one of the important cancers that actually coming in. And look at the mortalities, next slide. About 10 million people lost their life in 2020. And look, if there are developing countries like US and Europe, the incidences are very high. Earlier slide, if you look, their incidences were 25%, 35%. And their mortalities are low, by the way. We are in a developing country, thankfully. I don't feel that if I have to give an advice to Prime Minister, I will say that we should be, not poorer, but we should be in the current status. Because developing countries has its own problems. And they have been able to actually increase the survival rate far superior than the underdeveloping countries. The incidences worldwide averagely in a developed countries are 25% to 30%. India's prevalence number is about 10%. And let us look at the statistics what India has it. Maybe this is what actually when a cancer patient comes on the board, most of the patients, statistically 90% of the cancer patient when they come to the clinic already have a metastatic signature by this CT PET scan. That means they are not treatable. But whole pharmaceutical industry develops a drugs which can actually improve the overall survival, progression-free survival in metastatic setting, because these drugs are expensive. Some of them will be $1 million and they are not accessible to Indian populace as of now. But there's a one way of actually creating innovations, identifying mutations. Cancer is a bad luck disease, by the way. And all of us know that, the incidences in India are actually not accountable meticulously. There are biological studies over there. Remember, only 10% of cancer patients die because of a primary cancer. And 90% of patients die because of a spread of a cancer. However, by natural hemostasis and process, we do not get early symptoms of a cancer. That means when there is a primary cancer, suppose there is a lung cancer or a breast cancer, at an early stage, during that process of cell proliferation at a site which is a primary now of one organ, which is an epithelial cancer or a solid tissue cancer, at the same time, some of the cancer cells which are actually in a proliferation state have habit of detaching from the primary site. Nature has made it, reason for actually spreading the cancer. Because of cell adhesion molecule, less cell adhesion expression, these cancer cells fall. They come into the circulation with difficulty. 99% of them have very short half-life. They are eliminated by your immune system. But 1% of these fugitives, they start finding other tissue which is better substrate, and they start actually continuing to proliferate there. So the primary cancer initiation is actually a beginning of a metastasis process. Unfortunately, there is no way to capture these cancer cells unless you have a very intellectually, intelligently designed materials. And that is what we have been able to do it. The necessity of creating and designing chemical materials to capture this very less abundant cells is the challenge of material scientists can solve it. That is what we did. Next. So cancer has different problems. Let us see the terms: metastatic disease relapse, cancer dormancy, and on the other hand side, there is a strong term that is coming since last 2-3 years, minimal residual disease. So what does that mean? The scope and objective in cancer treatment is always to increase the overall survival. From patient's perspective, they want to be cured. But we say that when there is a cancer, it comes back. So what goes around, it comes around. Why? Because there are few fugitives that actually were in circulation, blood circulation. They can be dormant for years and decades, especially in the breast cancer of one type. The cancer patient actually, we hear that, is a survivor. But in a follow-up, after a few years, unfortunately, they come back with expression of different biomarkers or expression of other component, and that actually spreads either in the same tissue, in a micrometastasis or the distant organ. So therefore, the challenge is actually in a cancer is this threefold, and our technology is actually placed at these three important components of a cancer. And how, I will tell you. I think this was a statistics slide, probably missed it. India had 1.2 million new cases, and this number is rising, by the way. And we have head and neck cancer in females, breast and cervical cancer. Fortunately, there are only 2 cancers which can be physically examined or the test can be done. Unfortunately, rest of the cancers do not have an early detection. Only few companies that are actually coming for some early detections in other solid cancers, including lung cancer, by genetic testing. And they are going to come in a big way because they are now, multi-billion-dollar companies doing multiple clinical trials. But remember, early detection, if somebody finds that somebody will get into the cancer before the birth, that is what is the capability of today's genomic science. Or after the birth, or after 5 years, the oncologist and the board cannot treat patients unless there is an evidence of solid tissue biopsy by pathology. Confirmation comes by the structures of a cells that pathology sign, and second is by the CT PET scan or MRI, which is a confirmatory for the FDG or fluoro-D glucose sugar uptake. Without this, no cancer patient be touched. This is what is a universal guideline. So therefore, even if we have a proficiency or possibility of actually generating cancer, because there is a germline or somatic cancer in your family, you can actually get detected, but you have to continue monitor. So therefore, detection at an early stage is important, but there are no decisions for the treatment. You cannot take a treatment decision. What is more important, if I actually tell you today in a simplistic word, is detection of a spread of a cancer is more important in a primary cancer stages at the early one. And that is very, very challenging, and exactly that is what we do by our liquid biopsy blood test. Next slide, please. So I already spoken about there's a primary cancer. Some of the cancer cells detaches, they shreds. These positives are actually on the run. They are in the peripheral blood circulation. They find other substrate, and that is how we spread. The focus of treatment is only on the primary cancer. There is never actually a focus onto the secondary cancers when the patient come in the primary stages, stage 1, stage 2, early stages. This is a cancer circulating tumor cell from a cancer patient. If I look at a cancer cell, I think this patient is no more, in a survival. But we monitored and captured these tumor cells, which is actually those who like a biology. The cell is with CD45 negative, which is a biomarker present onto the leukocytes. There are billions of leukocytes per ml of blood. And there is a cytokeratin expression on epithelial cells, which is on a high regulation or upregulated cytokeratin in cancer patients circulating tumor cells. There is a DAPI nucleus tendi, and when you overlay this image, you get a qualified circulating tumor. The challenge in this technology is actually eliminate false positive and false negative, and that is why regulatory body actually takes so much of time, that you have to prove that not you capture the cell, but you have to prove that you do not decapture cell or you don't capture a cell. So your efficiency has to be 100%, and that's exceptionally challenging because, next slide, 1 ml of blood will have billions of other cells. Nature has actually created microparticles of different sizes, shapes, colors, and purposes. But nature has also created those kind of cancer cells which are in the circulation. Scientifically, role of circulating tumor cells, which is a telltale signature of a metastasis, has been termed as a seed and soil theory before 145 years before. However, it took 2004 for scientists to bring this technology in US with an approval in US FDA. See how much time that science actually elapsed. And the most important is that there are companies who are trying to create this kind of a sensitivity to capture these circulating tumor cells for diagnostics, for prognostication, and for surveillance of cancer patients who are in complete remission, or we call them as a cure, right? Because it hits back, and that is what is most important, that it is so sad not to detect the metastasis. And when there is a metastasis, there are no options. So those who are in chemistry departments, you have come by the pressure of Dr. Anand Kapadi or madam here. This is for you. We have a magnetic nanoparticles. We use carbon allotropes like graphene and CNT. Those who are in polymer chemistry, we use polyamidoamine dendrimers. We have enhanced the physico-chemical and biological properties of these components by creating a system which adds a unique functional capability. Right? If there are individual components, like for example, iron oxide is a paramagnetic component. But when it is with polymer, it has an enhanced hydrophilicity. When there is a graphene, it actually has a capability to add multiple other functional components, including antibody like anti-epithelial cell adhesion antibody. So what we did, we enriched these multi-components. Like a startup journey, you need a founder, you need an investors, you need an angel investors, you need clinicians, you need clinical expertise in this space. That's a multi-component story, and that's what is actually circulating tumor cells story. I'm coming to last 3 to 4 slides, please. So we run now multiple clinical samples. The blood samples are collected at the places including at Jammu Kashmir, Delhi, Bombay. There is an hub and spoke model where the blood samples comes to Pune lab, where we manufacture this kit, and every blood sample requires only 300 microgram of this material, which is enriched with an antibody. Now the beauty of this is our manufacturing batches is in the milligrams. And if we mess up this antibody conjugation, our scientists, who came for a job, will actually make us, outright out there from the technology. So it's a very important. We can't actually export this kits because the biological process of actually taking a blood sample, mixing these particles with the blood in 1.5 ml is an immunostaining process which takes about 4 and 5 hours. Everything goes onto the fluorescence microscope in the 96 well plate. All that what we remove. If there were cancer cells, we read this onto the automated fluorescence microscope where there is no human interface required. The machine does all job. There's an artificial intelligence. There is small codings over there, and it tells the size, shape, color, intensity of these cancer cells against leukocytes, against other blood component. And that is what we do. We have done now more than almost 3,000 tests. And I probably have a world record of looking every cancer cell for every patient, which is more than 26,000 today. There's no such record that anyone is scientist. Every cell goes through my eyes. And the moment I will sit in my car, there are reports actually has to be done. My pathologist need my help. My team needs my help finally. And that is what is actually interesting. Converting chemist to pathologist job or looking into the cell biologist based on the practice, based upon experience, based on actually what we have done. We now have another technology to isolate cancer cell, keep it live, isolate DNA, enrich the DNA, do sequencing at single cell with a whole genome amplification. Any company which can design material to capture circulating tumor cell and keep it live for genomics becomes $25 million valuation on day one. And we recently filed that patent. So 2 platform technologies over there. What we do now is actually we are developing an extracorporeal device like a dialysis to take out blood sample outside the cancer patient's blood and body and circulate through the device with 3rd generation materials which are more compatible. They are more non-immune causing issues, including the non-hemolytic aspect of it. And then we expect that the blood sample will go to the cancer patient so that the load of cancer cells, as we see sometimes, in 1.5 ml cancer patient can have 100 cells, 50 cells. 1 gram of a tissue daily releases 1 million cancer cells. Just imagine how much 7 liter of a blood might have cancers daily. So higher load is then shorter survival. And without a treatment, we hypothesis that the cancer patients will have a benefit. And we are the only active company in the world doing this in US today, with 2 US recently patented. So you start your journey somewhere, you actually continue to run somewhere. So it's not A to B point, it's from A to B to C to X, Y, Z. Next slide, please. I will come to the couple of cases. Go ahead and close my talk. So this is what the report actually looked, whether there's a cancer patient, the blood was collected, how many cancer cells we find, there is a one cell in this cancer patient. Unfortunately, my friend's wife who is now taking treatment here in Mumbai. There's also something called as a PD-L1 expression like cytokeratin you heard, and there is a real other protein which has been expressed on these tumor cells, that nature has created such programmed death ligand kind of a protein that immune system can be deceived by this cell. So that is what is the science that has come now. So pathologists can find this PD-L1 onto the tissues, which is a static biomarker, but now people have started realizing, doctor has realizing this is a dynamic biomarker, because if there is an expression of a PD-L1, there are specific targeted therapies, treatment decisions can be taken. So that's a second line or third line of a treatment that goes in. Simple functional assays. And in order to do this, US will charge almost $4,000. And we have done 90% free test in this country so far, out of our 3,000 tests that we have actually obliged to give it. And we will continue to do it. So this is on the right hand side. I think some of from this technology aspect, before we come, two cases only quickly. 50,000 rupees is the price. Every test we do, we are in the losses, by the way. Sometime, I ask my scientist, do you have a test today? I said 5. I feel bad because I am spending money over there. Your expenses are actually high. Why we do actually this? Because it's not a revenue model generating company. And I will tell you actually what is important. The turnaround time which I skipped is about 12 days in US. You take a patient blood sample, report will come 12 days. Here we do in just 2 days. Volume of a blood that companies require is 10 ml. We do this only 1.5 ml. 200 hospitals, around 200 oncologists, and that is what is a reach that your technology has actually easily been able to do it. And we hope that we will enhance this to 500 hospitals and oncologists in next 1 year of a time through our cells and team over there. Next slide, please. What we do is we look at the cancer circulating tumor cells at different stages and look and coordinate for the survival of this patient. And this study has been actually India's information, knowledge that we give it to the world, because head and neck cancer is not their problem, it is our problem. And you can predict the survival of that particular patient based on this biomarker, how long that patient will survive. You can take a decision whether there should be a surgery or there should be a neoadjuvant chemotherapy. It's a little complex, I will skip this at this point of a time. Very interesting case before I close here. 2020, all of us were in the dreaded corona COVID time. One of our oncologists I was speaking, Dr. Nirmal Raut from Bombay, who has actually taught us a lot of things in this. The patient, female 65 years, come to the clinic with a CA rectum. That's the staging. The treatment is like neoadjuvant chemotherapy with very standard drug, Folfox or Folfirinox. There is a chemotherapy and radiation therapy, that's a CT-RT. There is a surgery that was done in October. There is a maintenance therapy called as a Capox or capecitabine. And at this point of time, the patient is completely cured from this cancer. There is nothing, no burden by the CT PET scan. Absolutely, your case is closed. For some reason, this oncologist in COVID tried to make a fun of our test. Sends a blood sample to us. And we don't know the history of this patient unless, later on we try to grab it. 2 cancer cells were actually roaming in 1.5 ml. Imagine in 5 liters of blood how many cancer cells are there. Very shockingly, this is a panicky situation for patient because they read into Google, they know that the disease is progression against an information that oncologist has told that you are now disease free. Your status is disease free status. You are almost cured. Right? Today you go fine, celebrate. And that's what happened with this patient. Now the panicky situation starts. Jayant, how is sensitivity? Who gave you an approval? Who you are? Why did you find it? Right? And I should not have done this test. Even you are doing this pre-test. And for some reason, I said, where does this disease actually possibly progress into? A knowledge that we would not know. But, a curious question. He said, liver. And we just prolonged actually the MRI for this patient after 2 days. The patient had 4 lesions in liver. if any oncologist knows this, pathologist, they know that survival of this patient will not be more than 6 months of a time. So that I mentioned you that patients die because of a metastasis, not because predominantly by the primary cancer. This is what is an utility of these cancer patients, that happened. The patient was called for selective internal radiation therapy where the waiting period is 4 to 5 months. And the cost of selective internal radiation therapy in one of an hospital, you will not realize, is 10 lakh rupees. So we monitored this patient and we didn't find any circulating tumor cells there. This is what I mentioned, try to dare to say that we save patients' life through this test. So when we begin our journey, the journey was only for the materials, journey for survival, journey for a launch with the clinical trials. But you stumbled upon incidentally with these cases, which makes your life actually some with a purpose or some meaningful. This is what actually innovators can do through your startup companies, if I actually mention here. Next interesting story, and I close my talk. Sensitive content, those who can't really manage. Right? This is what we see every day. Look at a patient, female 80 years, again lockdown period, refused to do a tongue surgery. At 80 age, right? You don't want to lose a tongue. Simple oral metronomical treatment, 3,000, 4,000 rupees per month, continued with this simple treatment. So there is two ways or two kind of thought process, go for a surgery or go for the simple, tablets for this. The patient at her age, they just give a hope. We accounted circulating tumor cells in April 2023. 30, 40 days, you see what happened. The numbers of circulating tumor cells came. The prognostication of a treatment decision was actually well taken, where the patient actually didn't had to go for the tongue surgery. And in another 30 to 40 days, complete remission of this patient. And there is no clinical trial in the world that can actually show this kind of an implication into your own oral cancer. When I presented these cases to some of the leading doctors, including Dr. Klaus Pantel, they invite us to present this in Europe and other places. So that's the national competency that you bring through the technologies that actually you develop, and that is what is the power that we have it. So thank you, Anand, thank you, ICT, thank you, professors, thank you, students, for inviting me here today.
I think now we can open the session for questions. I think the presentation was so full of information, not just science, but otherwise. I think the way Jayant has presented all the details, even from the perspective of how, if you think about having your own startup, what all things which you need to think about, what all points you have to remember. So, but I would like to request you, if you have any questions, anything, any type of questions, basic questions, feel free to ask. Is there anyone? Yes.
Anyone with a bug of startup?
Oh. So with the facts which you presented about the same device, so I wanted to, just out of curiosity, the photodynamic therapy, which is also used as a part of a cancer treatment itself. So in the whole presentation, as per the perspective of how it is binding with the specific cancer cells itself. So in PDT, as per my knowledge, I know the most drawback thing about it is not finding the perfect cancer cell. Like, it cannot distinguish between normals, and photosensitizers are provided in the body, but they cannot distinguish between. So is there any scope of this material, like make it photosensitizer, and then it can only detect the cancer cell and act upon it, right?
The whole goal in a treatment of immuno-oncology, is actually have a targeted therapy. Like antibody drug conjugates, they spare the normal cells and they only are attached to the cancer cells which express some antigens. So that antibody drug conjugates are already available, in US, and last I think 1 year back or so, there were 11 such targeted therapies that were approved first time in US. That's a good news. In radiation therapy, limiting those components which are actually needs and exposure to light and make that kind of an impact. Nobody has ever been able to show that, but you need actually that photons with an capability of reaching to the normal cells. Nothing is impossible, right? So if I say that it is not there, that means there is a scope to do that. But you need to tag another component to identify those cells. Like you heard about CAR T therapy, right? It's about taking patient's blood sample, enriching those cells which were cancerous, develop an antibody, antigen against it, and send it. So this was the theory that Dr. Allison had thought it, and that therapy is already in the market, commercial. But currently, that doesn't happen. But radiation therapy is still very important, as I showed you. It can be focused to millimeter of a tissue today, sparing other regions. That's possible. I have seen in one hospital in Hyderabad very recently, real time, you could see a tumor with a CT PET scan, MRI parallelly, real time. Usually we see these still photographs, right? Real time there is a beam that goes in and the tumor actually starts shrinking in. So the technologies that what we have will not have a sensitivity to detect the tumor below 6-7 millimeter. I think I skipped this line, or usually that's a major content. That when we call a patient disease free, that means it is by the CT PET scan, which has a limitation of 8 to 9 millimeter of a sensitivity. That's what actually patient and doctors feel the patient is disease free. But the real is you should do a blood test. If your blood doesn't have a CTC, then you are a disease free. Or your blood doesn't have a ctDNA, cell-free DNA, then it is actually disease free. But in radiation therapy, there is, I think, many people have wanted to do this, but I'm not an expertise in this area. But there is absolutely a scientific objective that must be, carried. And your generation is to do that, right?
Any more questions?
So we talk about detection of CTCs. Is the population of CTC in the blood depending on the size of the tumor? Because if it is a very small tumor, it may not have as many CTCs.
So larger the tumor, higher the number of a dissemination of a cell. That's a theory. Like 1 gram of a tumor can actually shred millions of a cancer cell.
Yeah.
But as I mentioned, there is a theory when there is a primary cancer with say 100 cells, 1000 cells, 10,000 cells, one cell. There's a process of actually shredding these cells. That happens on a day one. Okay. So how fast, it depends upon each individual, depends on immune system, depends upon type of a cancer, depend upon the stage of a cancer, like are there lymph nodes involved. So TNM staging is actually tumor size, N is a lymph node, and M is a metastatic signature. That's how the staging is done. So higher the stage, higher the number of cancer cells have been founded. So that is already proven. We find often that the patient has a stage 1, but they might have more cells, and the whole world is about personalized precision oncology today.
Yeah.
So, but the clinical trial, they are stratification of 100,000 patients, and it gives you, some bars over there. But now the science, see, lung cancer stage 2, it depends upon the location of the tumor in that large lung. So if there are sometimes actually we find that the stage is very high, but we don't find the CTCs. That happen only, 5% times.
Okay.
But often we find cancer cells by more than 60-70% of time it is corroborated.
Okay, that's a good number. And the second question is slightly tricky. Normally, when an innovator starts with some startup, and then it reaches a particular stage, and then it is kind of it is felt that it should be merged with bigger companies or something, and then the innovators can move on to something else that they have been doing or they would like to do. How do you see yourself in this situation? Would it be the other way?
So I also wanted to do, this is what is the typical progression of any startup. You start up proof of principle, you have an IP, you get an venture capitalist, you have an, valuation of your companies to the next level. Probably, look here, valuation you ask, right, for the curiosity of Ananda. So the transformation goes on, but my angel investors also continue to ask me, if we fund you, are you going to actually exit? I said, no, I will exist. I will continue to do, because if the goal is to pass this to the lower strata of society, you cannot actually exit. Right? Therefore, it's a philosophy that you build on. So we don't actually exit for several years. We want to do this for at least 50,000 patients and then think of, until we are capable. It becomes very complex later on. Supply chain, more hospitals, machines, validations, audits, it becomes, so that time actually the founders, they want to exit, with some good funds of it. So if you see that we are now almost 200 crore rupees valuation company. We don't have that much revenues, but we are probably poorest liquid biopsy company today with the technology that what we have it. And we feel proud of it, right? Then second companies will come and have a joint venture. We get more richer in valuation when you have a joint venture. So the second company is about $20 million valuation now. This company is hoping to go to $100 million in another 3 years. So, it's all, a single cell has ocean of information, we call it. OneCell is the name of
and there is more than one correct way of going ahead.
So now what has happened is, fortunately or unfortunately, I have transformed into a person who can seriously bring something in the market by setting, operating. I am a very bad manager. I don't have any MBA degrees. But people trust that I am serious and I want to actually bring a technology in the market, launching a technology. That becomes your goal. Some people are good to propose and get money and exit it. Right? So this form of a science is, therefore it's a challenging to remain there, and therefore this is a golden cage to be an UDCT professor, right? It's a nice, you can actually, play some match and come in back. But this entity is entirely different, where you have everyday issues, everyday challenges, demands, right? And you have to go to the doctor. One of a doctor, I don't want to name, very most humble. When I did a test for this doctor, ma'am, this is for you, 15th August 2021, I get a call from one of the finest doctor that this country has. First said that, this is my name. Who are you? I said, my name is Jayant. Why did you think my patient is actually not in a remission? I said, you prescribe a test, I find the cell. If you feel that you are a god and write that certificate, I am fine. But my certificate which I give can actually defend any technology, legal battles. I am saying your patient is not in a remission. If you are fine, you just go ahead. And he was a very high profile patient, by the way. He didn't want to take a chance. Then he asked me that, what should I do now? I said, I'm not an oncologist. I'm a diagnostic company. You told me who you are. I'm nobody. I'm just a diagnostic company. You take a treatment decision because you are next to God. Right? So that kind of a tussle happens. And today, that patient is actually survival. He gave a continued chemotherapy. Now the patient's cells number are 0. And we have done, I think, 14 times this patient. And I will tell you, this patient is very, very important person in this country. I might mention that I want to go to the reach, reach to the, not that RICH, this is REACH, bottom of pyramid. But we have also catered the topmost important people in this country by our test.
Hello, sir. So regarding that sample size is 1.5 ml. So like the concentration of cancer cells will not be similar in like whole blood. At least at the first stage it will be very less. So like is there any possibility that person is having some cancer cells in the body, and like because we are taking only 1.5 ml of the blood sample that it is not able to detect. So that I was thinking if that sample size is decided considering that like.
So very good question. Thank you very much. This is where actually your sensitivity starts. So when you start a clinical trial, you are given or you take patients who are actually obviously metastasis. So you take 100 patients who are actually with a large size of a tumor. You look at the sensitivity that goes in there. So that's what is the first benchmark. Now when you take 3-stage, second stage and early stage, typically blood is heterogeneous, right? It's a bloody blood. Blood is bloody amazing thing there, okay. So cancer cells distribution in 1.5 ml is good enough distribution itself. So what you do, you take say 5 ml of a blood and do a triplicates of it. So second validation comes like this, like you blind your scientist with triplicates of 1.5 ml, and if you find one of a sample had a cell and 2 samples didn't had it, then you are gone, right? You should have 3 of the samples with same number, and that number has to be plus and minus 1. That is how the sensitivity is actually detected. You understood? So if you do only in 1.5 ml, so we know that our 99% of the time we don't miss a cell because there was 1.5 ml against another 1.5 ml and another 1.5 ml. So that's how you decide the volume of a blood and the sensitivity of a test.
Sir, and like one more thing, most of the first stage cancer cell, they have not not much prominent symptoms. So like people have tendency to avoid small health issues, and so it is a very big challenge to reach out to the people to take the test at like first stage or at the very beginning.
We thought
stage.
this, thank for that comment. We thought that our test actually is for those patients, early detection or early primary cancer. Now we realize that India has more than 50 lakh cancer patients burdened today. They are going to come back. They are going to come back for the metastasis. So the surveillance of cancer patients is more important who had a high burden of a disease, and they were lucky to have a detections early, and they responded very well. So it's a twofold. Don't allow stage 1, stage 2 cancer patients to migrate into the stage 3 because they were considered as a disease free because of an absence of a CT PET scan, which has a lower sensitivity, right? And that sensitivity must have a tissue of 10 raised to power 12 number of cells. So that's a larger cells that you capture. Here you capture a single cell which is about 10 ml, 20 micron or 10 micron size. So it has now progressed into utility. You can do a surveillance, you can actually prevent, you can detect, you can manage, so variety of capability, but it is not pan cancer. Colorectal cancers, I will tell you what happens. The treatment in colorectal cancer, which is now number 5th or 6th cancer in India because of food style and sedentary lifestyle. There is always a debate in colorectal cancer stage 3 whether the patient should be treated with 3 months of a treatment or 6 months of a treatment. So there are interesting clinical studies have been done where they found at the baseline number at second time point, third time point after 3 months, 6 months, the CTC number actually drops down whether the patient were treated for 3 months or 6 months. They look at the genetic load too with a cell-free DNA. But later on what happened, the patient who got only 3 months of a treatment against 6 months of a patient develop a resistance clonal colonies from the cancer. So it is very important that the treatment decisions 3 months versus 6 months based on the numbers and the monitoring can be also decided. That's how the clinical science actually improves by the evidences that you bring in.
So your test detects carcinoma. Can you do also for sarcoma?
No, this is, see, 80% of the cancers are actually solid tissue cancers. Okay, adenocarcinomas and carcinomas. It cannot be done for melanoma and sarcoma and blood cancers. AML, CML, this is not there. Because the cancer cell has to be from the epithelial origin cancers. So 80% of cancers can be suitable for this test. You got the answer? No sarcomas and melanomas. No. Which one? No, sarcoma is your liquid cancer. melanomas and sarcomas will be of that kind. Like your glioblastoma, brain
Okay.
cancer, there is a brain barrier. So CTCs will not be important. But CSF, cerebrospinal fluid causing diseases, definitely would have a possibility of a circulating tumor cells.
So can you test it also in CFS?
Yes,
That's possible.
right.
Thank you.
But the stability of cells in CSF is very less, so patient has to be admitted very close to our lab, and then we have actually such kind of situations. Patients actually fly from very farthest part of India on request of doctors in here Tata. We want to actually bring them to Pune, take a blood sample, run it fast to us so that you capture those cells. So very interesting science coming up. This is
Thank
just a
you.
beginning after 140 years.
Madam.
Any more questions?
Nothing on the startup, only on the science.
I think they are more concerned about exams which are going to come in next few weeks.
Thank you.
I think I would like to thank Jayant, because what we were expecting from this platform, that it should be a very open session, open discussion. He talked some part of science, which is very important because it was pretty exciting, something which has not been done, something which is very close to what we are. Maybe I have to ask one question, maybe if I can.
Yeah, please.
This is from a general point of view, that we are, when we look at ourselves, we commonly say that we are fit. Now we have worked in cancer, although it was not your direct field. But now when you have seen all these patients, and must have been from various strata, and do you think that this is something which is being expected out of us, that every 6 months that we need to have some kind of detection of whatever is happening to our body, which is a simple blood test or so? What do you think is more important? Because cancer initiation and then proliferation is something which cannot be predicted. You said that it could be possibly because of the lifestyle, and lifestyle nowadays is not something which we can really have a control over. So what would be your suggestion?
So the current era is actually detect and treat. Okay, detect and treat. So yes, you can predict now. Algorithm, AI, ML systems are coming very powerful. The ability to capture cells at one cell is, I think I forgot the most important statement of my talk. I'm so sorry, because maybe I got up 6 o'clock in the morning. So I will have an excuse of losing actually most important. Presence of one circulating tumor cells is associated with the worst outcome than the absence of a circulating tumor cells. So your question is actually, can we predict it and how fast? Yes, algorithms, they are able to do it. First, AI ML is very powerful now. And in order to detect cell-free DNA, which is your DNA coming out, there is a DNA that comes from the normal cells which are in aberrations, which are actually every day they are dying, and you get a DNA. Now, if there was an initiation of a cancer, there are a couple of companies like Grail who are doing it. They can take healthy people's blood sample as an early prediction or detection, and they can detect that ctDNA, which is different than the normal cell-free DNA. And what it requires that pull up a blood sample, non-invasive technology, but it needs a capacity to amplify that one DNA that came through the PCR in last 10 years. Now, we don't use PCR in our genomics company. We create libraries to isolate that single DNA, because there are actually codes, codons, funny components, chemistry over there. It can actually, go and attach to that DNA, and this DNA can be amplified. Now, the data that what you generate through this library of panels that goes into the sequencer has ability to run the sequencing within 48 hours. So you can take one drop of a, small, what we call it as an enrichment from 40 patient sample as an one drop, put it into the sequencer, and each of our DNA has a different code of a DNA component over there. And then it is amplified. The data that generates is 20 GB. Now, AI system can cure that data by 0.0001%. And that's how your tier 1, tier 2 mutations comes in. So, you are very close now through these companies in US and NHS, I think UK, they have started a new study. They are monitoring patients in lakhs today in UK and US. And they found that 40 to 45% of patients develop colorectal cancer or particular cancer in 2 years of a time. The sensitivity is now 40%. So it's coming in any form post 25 years. This kind of test, which will cost maybe $5,000, they are actually on the road. If they get license, we are also going to come into this picture. But doing that requires billions of dollars. Yes, it's possible to predict. It is possible to detect, and it is possible to treat. So we are adding that personalized precision prediction information in coming years.
Thank you again for a wonderful talk. I think this was the starting point. It was the initiation of our innovation lecture series, and I'm sure you all will agree with me that it was not just innovative, but it was so much substantiative with respect to the information that was given to us. So thank you very much, Jayant, and I would like everyone to applaud. Thank you everyone for patiently waiting for almost 2 hours, listening to the discussions which have ensued. I would like to now request you to have some refreshments which have been kept outside. And this is going to happen every 6 months. So we are going to scout for again someone who is innovating, coming out with solutions which is helpful for humanity. And this is a 3-year project. So if you have any questions, further questions which you want to follow up with, or you want to maybe interact with Jayant, I would request Jayant, maybe if I can share your email address with them, and then please get in touch with him. I have already visited his fantastic laboratory in Pune. I would request you just go on to this website, OncoDiscover, you'll get all the details. And if you have more questions or you want to visit, send an email. I'm sure that he will be more than happy to show you around. And if you are those crazy minds, what he was talking about, which he supports, not now, maybe after your PhD, postdoc, and then maybe you think that, not that you want to be like us who are scientists, as we really very nicely mentioned, that we are stuck here. But no, if you are of that mind that you want to do something new, something very innovative, but you are kind of concerned about money and all, please do get in touch. Okay, so thank you all, and we look forward for the next talk, which should be in next few months, 4-5 months or so. Thank you very much.